Donc, bonjour à tous. Nous avons le plaisir d'accueillir aujourd'hui Malgorzata Gazda, qui est actuellement en postdoc dans l'équipe DYOGEN, le groupe DYOGEN, sur l'évolution moléculaire de la menstruation avec des assais génomiques comparatifs et fonctionnels. Avant ça, donc, elle avait commencé par un master à la Jagiellonian University à Cracovie en Pologne sur la corrélation entre fitness et hétérozygosie, et une thèse à l'université de Porto au CIBIO sur l'utilisation des NGS pour l'étude des traits génétiques qui sont importants chez les oiseaux, comme la coloration, le chant, la performance de vol et l'orientation chez les pigeons. Et donc, je vais lui donner la parole. It's up to you.
Thank you so much for your kind introduction, and I'm very excited to be here and share with all of you my PhD work. Just sharing my screen. Do you see it in the presenter mode?
Yes.
Okay, cool. Well, already my name and my affiliations were introduced, and if anyone is interested in my research, want to get in touch or discuss or collaborate in the future, please feel free to reach me by Twitter or my email. Yes, okay. So in general, in nature, we have a big diversity of different phenotypes, and these different phenotypes can be shared across different species. Like here you can see different species of blue butterflies, but in some cases you can also have a big diversity of different characteristics, different traits within one breed. And here you have an example of dogs which can be small, big, and this phenotypic variation, which we can see as in the dog in domesticated species, it's useful to study many different things. So from the traits that are important for humans, so for example, meat or milk production, through medicine, and ultimately, which is very exciting for me, we can also get insights into evolution and genetic basis of traits. Birds are particularly a good model for studying many evolutionary questions, because they display an amazing diversity of different traits. So here in the picture you can see a really great variation in different colors. And other very exciting examples of traits in birds is the flight and athletic performance and the song. But today I will focus on the coloration. And during my PhD, I chose the domestic canary as a model for my studies. And that was really good choice, because it is a really good model, since the breeders for many years selected canaries for many different traits, starting from coloration, and that's what I tried to figure out, but also through song, different feather characteristic, and other traits, which some of them I'm also still trying to figure out. And of course, it's easy to breed them, it's easy to keep them in the lab, so it's really great model. And the objectives of my PhD was to try to find the link between the traits important for avian evolution and their genetic basis. So I asked myself a couple of questions. So first, I wanted to know if the genetic basis of studied trait are simple or complex. Then I wanted to know what gene or genes are involved, and then what kind of mutations are affecting those genes. Is it coding or regulatory variants? And how this genetic variation evolved. So was it the selection on standing genetic variation, or maybe de novo mutation or introgression? And finally, and really excitingly, what are the biological implications? What can we understand more, making this link between the genotype and phenotype? And maybe trying to put this into the context not only domesticated species, but also it gives a further opportunity to study those genes and phenotypes in the natural populations in the wild. So to do that kind of projects, I had 4 different kind of parts of the projects, different approaches. So first, very important, was the phenotypic characterization. So I had to choose the model and then characterize what is the difference between those 2 breeds. So here we have the example of yellow versus white birds. So to do the phenotypic characterizations of them, for example, I would measure the carotenoid content in the feathers to understand how much carotenoid they have in the feathers. And then we would take the samples from the birds belonging to different breeds and perform the whole genome sequencing and compare the population differentiation, genetic differentiation between those 2 breeds to find out the regions that are the most divergent, and then do a bunch of different analyses to try to understand what is the variant responsible for this phenotype. And once I had the candidate mutation, the important part was to try to find out like how to do the validation. So for example, to clone the specific variant into the cells and then confirm if there is the activity of that cells or not. And finally, try to link the characteristic of that breed with different biological implications. So first, I studied those birds that I already mentioned, so the white recessive bird. So the idea was to understand what gene allows bird to acquire carotenoids. So here in the picture you can see the white bird, and they don't have the carotenoids in the feathers, and you can see the yellow bird, and they have the carotenoids in the feathers. And that is the example of the data set that we used. I will present it only for this project because it will be similar for different projects, but just to give you an idea like what kind of data we used and how we proceeded. So as I already mentioned, we did whole genome sequencing of canary pools, and then we did also the classical genetic analysis. So we actually crossed the birds and did the linkage analysis to confirm that indeed this mutation segregates as it should, and so on. And we did the genotyping for IBD mapping to narrow down the region that we find with the genetic scan. And once we had all this polished, let's say, we proceeded to functional experiments. Some of the functional experiments were actually the part of the phenotype characterization as HPLC here in the feather, skin, and other tissues. But then when we had the candidate gene, we could measure the expression in the specific tissues during the qPCR. And if that was the coding mutation, we could also do protein structure modeling and the cell culture experiments, as I mentioned before, for example, trying to quantify carotenoid transport in the cells. And he, oh, sorry. So first, as I said, we sequenced the birds. We looked at white versus white recessive versus yellow, and we looked across the genome. Here we have the FST statistics, which is quantifying the population differentiation between those two pools. Well, you can see some peaks across the genome because the thing is that there are many different traits that the canaries were selected for, and the breeders also cross different breeds all the time. And you might also get some peaks, for example, because the individuals could be a bit more related, especially in case of some more rare breeds. So that's why we needed this crosses and also the IBD mapping to make sure that this is the right region. So the most differentiation region here consists the SCARP1 gene, which is scavenger receptor class B member 1. So we concluded that that was a good candidate gene for the wide recessive phenotype based on the population differentiation. And then what we did next, we characterized different isoforms of this gene, and we found out that the yellow birds had only the wild type gene, so having all 4 exons, whereas in white recessive there were 3 different types of isoforms, but the most abundant was the isoform 4, and in this isoform 4 there was a splice site mutation in the exon 4. So what was happening, it was the isoform was missing a large portion of this exon. And then to understand what happens if this exon is missing, we did the cell culture experiment. So we cloned different isoforms into the fibroblast cells of chicken. And then we put the cells into different media with either chicken serum or zeaxanthin. And then we could observe if the cells absorb carotenoids or not. So we quantified the carotenoid concentration in those cells. And here you can see that in the yellow cells that had the wild type isoform of SCARB1, they had quite a lot of carotenoids. Whereas looking at isoform 4 and also control, that was fluorescent protein only, we see like the trace amount of the carotenoids. So we concluded that basically the cells that had the wide recessive isoform, so this isoform 4, they didn't uptake the carotenoids. And the implications from this study was that, well, SCARB1, scavenger receptor, is the essential mediator of the expression of carotenoid-based coloration in birds. And we also showed the potential link between visual display and lipid metabolism, and that is why, because SCARB1 gene is also involved in the lipid metabolism and all the cholesterol pathways and so on. So that's how we could potentially link this lipid metabolism with coloration, but this requires a bit more studies. And finally, SCARB1 is the Asian and conserved mechanism of carotenoid uptake in animals, and this is because the orthologs gene for SCARB1 were shown for example, in studies on Drosophila. So they have the similar function in birds as well. So now we know how the carotenoids are absorbed, but then there are many different ways that the birds can differ in the carotenoid content. And my second study is about the carotenoid ornamentation. So here we chose two different breeds. One is the Urucum canary, so in other words, red-beak canary. So you can see in the picture they have intensively colored red beak. And we compared them versus the red canary, and that is because, first of all, we didn't want to pick up the variation that is responsible for red coloration, so it was easier to have both breeds red color instead of yellow or white, for example. And then the second reason was that well, this breed arose in the colony of the red birds as well, so that was easier to study. But interestingly, there is also the other color variant of this breed, and it's yellow red beak. And we also checked those birds, and actually the story is the same, so they have the same mutation. But I'm gonna show you that in a minute. So again, we looked across the genome and we found the most differentiation region in the genome. And that is the BCO2, beta-carotenoid oxygenase 2 gene. And then this gene was implicated for carotenoid coloration in many different cases, and we are getting more and more of them. So before this study, there were examples from other domesticated animals. For example, it was shown that it regulates the color of fat and meat in different species, like cows and sheep, I think. And then we looked again for the mutation. We found out that there is the missense mutation in the red-beaked birds. And this made a lot of sense because those birds actually were a bit difficult to breed because they have some visual problems, some reproductive problems as well. They didn't really breed that well. And it seems so you can see here the protein part of the protein sequence, and the region where we found mutation is quite conserved across different species, and basically this mutation only appeared in those red big birds, and we also did some protein modeling. So you can see here that the change of amino acid of arginine to histidine caused the structural distortion of the protein. And that distortion leads to the observed loss of activity. So basically, what is happening in those birds is that the BCO2 it's not active, therefore the carotenoids are not disrupted, not removed, so they accumulate BCO2 in all the tissues. Oh, sorry for this. And again, to confirm those results, we did the functional validation. So we cloned the BCO2 again to cells, and we did the assay that. Well, so in the wild type BCO2, we had the cells with the carotenoids, and then we checked if the cells were metabolizing the carotenoids, basically. So in the wild type, which is in blue, you can see that there's no substrate. But there are apocarotenoids, which are the product of the removing the carotenoids. Whereas if we look at the mutated BCO2 or denaturated BCO2, we see a lot of substrate, but we don't see any apocarotenoids. There's no product of the metabolism, so they basically don't don't disrupt the carotenoid, don't metabolite them. And then the implications of this study is that carotenoid cleaving activity leads to coloration gain, modification, or loss. And we also looked at the retina because the coloration genes might be important for the cone pigmentation and therefore the sensitivity of the vision, and therefore and we also found the differences between the red regular birds and this mutants. So that's how we concluded that BCO2 impacts cone pigmentation and tune sensitivity of photoreceptors. And finally, we found out the interplay of the signaling benefits and physiological cost may be mediated by regulation of BCO2. And the final story is the study that was published last year in Science, and we looked at the very exciting phenotype, which is the sexual dichromatism. So the male, you can see it's flashy. It has much more carotenoid coloration than the female. And to do this study, we took advantage of the model that was created by the breeders. So what breeders did, and that was actually the process of selection for the red canaries, because the breeders for some reason wanted to have the red birds, but a byproduct of that was actually creating the mosaic canary breed, which is really beautiful too. So how did it happen? It was the cross of the red siskin. You can see the male and female here with the common canaries. And then we got the hybrid, which of course looked like something in between. And those hybrids were back crossed with the canaries. And then finally, we got the canaries that had the different carotenoid content in the feathers that was introduced in the genome from the red siskin. And that is the unique model to study dichromatism in finches, because normally it's very challenging to study this kind of questions because the sexual dichromatism is usually fixed in the species. So they either have it or don't have it. It's and then the species are of course more divergent than having the breed of canaries being sexual dichromatic or not. And again, we look across the genome, and here I'm showing you two different statistics. So we are already familiar with FST, but there is the FD statistics, so the fraction of the genome shared through introgression, and you can see very strong peak in the same region where there is the strongest peak in the FST2. So this second statistics, it's quantifying how more similar the birds are to either the one parental species. So in this case would be the canary, or the other one, and that would be the red siskin. And you can see here that the most of the genome is very similar to the canary genome, but then there's this region which is highly divergent, and that is because it is introduced from the siskin genome. In FST, you can see again some smaller peaks, and there is one significant peak, and that is the region which is responsible for the red coloration, and that is why, because we used different colored breeds, so we didn't use only one colored breed, we used many of them. And this, so I mentioned already before the red canary process of selection, and this was the same story. So the red was introduced from the red siskin, and therefore this gives a very strong signal in the genome. That's why we could see it here again. And then Yes. And then to narrow down the region, we again did the SNP genotyping in the region of interest, and we genotyped the siskin and then other canary breeds, and finally the mosaic breeds. And we can see that in the siskin, all the SNPs in the whole region, 1 MB, are homozygote siskin, and the same is true for canaries. They are all homozygote canary. But here you can see why this high resolution mapping is very useful, because many of the SNPs will be either homozygote siskin or heterozygote. But there is one small region that consists 3 genes, BCO2, PTS, and TEX12, and all the mosaic individuals were homozygote siskin. And therefore, well, again, BCO2 is a strong candidate gene for explaining sexual dichromatism. And we tried to do some experiments again. So first, we measured the expression. So we found out that the females express more BCO2, and then they have the less coloration than the males. Then we didn't find any protein coding variant and other any other coding variants. So we hypothesized that this is likely cis regulation. So we took the carrier individuals, so the individuals that had both the siskin and the canary allele, and then we quantified how many reads per individuals they had, and we found out that the most abundant, so they should have 50-50, but they had a strong bias towards siskin allele, so therefore it's likely to be the cis regulation. And we also did in situ, and we confirmed that BCO2 is indeed important for having or not having pigment in the feathers. So in pigmented follicles, there was the pigment, but there was no BCO2 expression, whereas in unpigmented follicle, there was no pigment and there was a strong BCO2 expression. So the more BCO2 expressed, the more it's active and the less carotenoids, because they are removed from the tissue. Well, sorry for my mouse. To put this study into a wider evolutionary context, we also did the transcriptomic analysis in different species, and we chose in wild species. So we took common canaries, but not the domesticated one, but the wild ones that we can find in the Canary Islands. And then we took also the sister species, which is European serine, and then closely related other finch that has strong sexual dichromatism. And then we did RNA-seq, as I mentioned already, in 2 different patches in the canary body. So we sampled the breast and the belly, and different colors here tell you how divergent are the regions. So, for example, in European serine, in both patches, so in breast and belly, the males have a lot of coloration, whereas the females don't really have. So there is a big difference between them. And those numbers in the circles are the differentially expressed genes. So we see that when we have a big differences in the patches, there's lots of differentially expressed genes. Whereas in house finch, there is a big difference in the breast, but if we look in the belly, then the males also don't have coloration. Therefore, there's, well, no difference between those two regions, and we found really little 8 differentially expressed genes. So we concluded that gene expression divergence between males and females correlates with sexual dichromatism. And the implications of this study are that the dichromatism in birds can be generated by a simple molecular mechanism that is driven by large effect gene, in this case BCO2, that the function of this gene exert in the periphery tissue, so it's, well, tissue specific, and then carotenoid signaling might be shaped more by cellular processes than resource limitations. And with this, I think I already showed you that the domesticated species have a great power to study the genetic basis of traits. And there is a big diversity of traits and evolutionary trajectories. So there are many different mechanisms that the trait can arise, and different types of mutations and regulation, how this might work on the molecular level. And identifying those genes correlated with the specific phenotype helps us to understand the questions in general biology, and also allows us to ask further questions more on the individual evolutionary well, fitness level. So I would like to thank all my colleagues, my supervisor Miguel Carneiro, my funding for my PhD, which was from the Portuguese Foundation for Science, and I will be very happy to take your questions. And thank you for your attention.
Thank you very, very much. Who wants to ask a question? If no one wants to start, I would have one. What is the divergence between the species, the European serine, the species you presented for the last example? Are they very closely related, or are they more?
Yes, they're very closely related because they are considered to be a sister species.
Okay, so for that kind of things comparison, you really need things that are the species that are extremely closely related to be able to do really meaningful.
Yes, because otherwise you will get thousands of differences, which you might not be 100% if it's related to the phenotype or related to divergence between the species or many other things.
Of course. Thank you very much. Someone else? Bertrand?
Est-ce que vous m'entendez?
Ouais.
D'accord, j'ai dû enlever mon casque, le micro passait pas. Yes, I have a question about the Raccoon mutation, the red mutation in BCO2. Is there an impact on the skin color? Because in chicken, BCO2 controls the skin color, not the feather color. So maybe, I don't know if the skin is colored somewhere in these birds.
Yes, that's a really good question. So I explained you about the difference in the beak. But yeah, if you mute yourself, maybe it's better because I can see the hole. But there's also the difference in the leg color. So basically that regulates the coloration of the bird parts. Mm. I don't think the skin, like in the belly as well, but like in the legs at least you can see a very big difference. Maybe if we would have removed the feathers from different regions of body, there might be some difference as well. But it is basically, well, it affects not internal organs because we checked that, but for example, as I mentioned, it affects also retina, so they have the different cones.
I can have another question about the carotenoid pigmentation.
Sure.
It is supposed to be a good signal for male fitness, for example, in blackbird for the big coloration. Do you expect something like that in all the finches you have studied about the chromatism?
If I expect what? Sorry, to
If
see
you expect that the yellow color, the brighter yellow color in males to be a kind of good signal of male fitness, male health.
Yeah, that's an interesting question to study now, because we sort of so we see more and more BCO2 to be showing up in different studies. So it seems to be, well, there is other gene that also mediates the sexual dichromatism as well, and the red coloration, which I already mentioned in canaries, and that is CYP2J19. And if you look across the studies, at least in birds, it is showing more and more for different species and different things in nature. So from my study, it's really hard to say like how does it correlate with the individual quality signal. But well, it seems that it might have been a little bit different than we believed, because so the idea of the individual quality signal with the carotenoids is that the birds cannot produce carotenoid themselves. They have to acquire them from the diet. Yeah. So it's supposed to be an expensive trade. But here we can see that, well, both males and females have the carotenoids. The thing is that actually the females are removing the carotenoids. And I think this is also everything about the balance between the carotenoids and carotenoid metabolism, because there were also some studies that showed that having too much carotenoids is actually toxic for the birds. So they showed the examples of some problems with the muscles when having too much carotenoids. But it is also a little bit similar in our case too, because those red-beak birds that have a lot of carotenoid content, I already mentioned that they had some health problems. So I think the balance is really needed, but I will be very happy to see in the future follow-up studies more on the ecology and individual fitness and how does it correlate with BCO2 and all the theory of signaling, honest signaling. So I think there's a lot to ask still.
Someone else? Well, then thank you very much